Very good morning to all my dear students. Today we will discuss the third session about the auditory physiology. Today we will discuss about the auditory pathway in details and also about the neural processing of the auditory information. As I had said in the last class that May is a better hearing and speech month. So let us talk good and speak good and spread more positivity. So coming to today's learning objectives. Today we would see the auditory pathway details. Coming to next, the features of the auditory pathway. Then some information about the auditory cortex. And then the neural processing. Last time we had seen the inner ear. So this is a cochlea which plays a very, very important role in hearing. The cochlea on the cross section is divided into three separate compartments. That is scala vestibuli, scala media, and scala tympani. The scala media houses the organ of Corti, which is a very important structure responsible for the transduction of auditory information into electrical signals. Coming to cochlea and the organ of Corti. Now, in the details, we see that this is an inner hair cell comprising only a single row, whereas these are the outer hair cells which are present in three or more. The supporting cells of diatel, they offer anchorage to the hair cells, and this is the basilar membrane. These are the efferents which carries the information towards the brain. Now, to see what happens when perilymph is set into vibration. So, first step is striking of the tympanic membrane, which is the sound is conducted via the ossicles towards the foot plate. Now, foot plate is in contact with the oval window. Now, in the scala vestibuli and scala tympani, we have perilymph, whereas scala media contains endolymph. So, depending on what is the frequency, that particular area of the basilar membrane is set into vibration, which matches the natural frequency of the sound. So, for here, the sound will travel here, and it will cause movement of the basilar membrane. So that area would get pressed, and this pressure is then reabsorbed by the round window. So coming to stimulation of hair cells by each frequency. Now, as each section of the basilar membrane has different mechanical properties which attributes to the different hair cells respond to different frequencies. For example, if 4 kilohertz sound is coming, this will stimulate the hair cell which has got that particular natural resonance. So the auditory nerve fibers are activated more easily at natural resonant best frequency. So according to Helmholtz resonance theory, the characteristic frequency of an auditory nerve fiber reflects the resonant frequency of the basement membrane from which it originates. So coming to the relay stations for the auditory pathway. The last time we had seen that we had I hope you all are familiar with these names, that is the cochlear nucleus, superior olivary nucleus or complex, and inferior colliculus and medial geniculate body. So coming to spiral ganglion. The spiral ganglion, it starts from the hair cells. So, spiral ganglion houses the cell bodies of the first order neuron which arises from the hair cells, which carry information from the hair cells and travel within the osseous spiral lamina. So, this spiral ganglion, it will comprise the cochlear branch of 8 nerve. So, when it joins the vestibular nerve, it forms vestibulocochlear nerve. So coming to the first order neuron. So the first order neuron arises from the nerve fibers of spiral ganglion, and it will terminate in ipsilateral dorsal and ventral cochlear nucleus, which is located in upper medulla. Now coming to the cochlear nucleus. Now cochlear nucleus is divided into ventral portion and dorsal portion. So here you see in this diagram, this is the dorsal portion of the cochlear nucleus, and this is a ventral cochlear nucleus. So the dorsal, if you focus on this image. From the dorsal cochlear nucleus, most of the fibers, they cross the midline and reach the lateral lemniscus. Very few fibers go to the ipsilateral lateral lemniscus. So from the dorsal cochlear nucleus to the lateral lemniscus, remember this thing. Now, when you go to ventral cochlear nucleus, if you look in these diagrams, look at these green structures, these are nothing but the superior olivary complex. Now from the ventral cochlear nucleus, few fibers you see, they are reaching the ipsilateral portions and few fibers go to the contralateral portion. So in the ventral cochlear nucleus, most of the fibers, they cross the midline and reach to the contralateral superior olivary nucleus. Okay. Now, these crossing over, this decussation forms trapezoid body. So this is what is trapezoid body. So trapezoid body is nothing but the fibers which are crossing from the ventral cochlear nucleus towards the contralateral superior olivary complex. So we can say that this trapezoid body is located just next to superior olivary complex. So remember this thing, the trapezoid body, which is in connection with the ventral cochlear nucleus. Now next. So we can say that both from the dorsal cochlear nucleus as well as the ventral cochlear nucleus, most of the fibers decussate and few do not. So information from both the ears reach bilateral lateral lemniscus. So if there is a lesion above this area, means beyond the cochlear nucleus, it will not cause any serious hearing impairment because information from both the ears reach both the lateral lemniscus. If there is a lesion before this cochlear nucleus, it will lead to unilateral ipsilateral deafness. Come to the second order neuron. Now, the first order neurons had terminated in the dorsal cochlear nucleus. So the second order neuron will arise from the cochlear nucleus, few fibers crossing, few uncrossing, ultimately reaching the superior olivary complex. Now, the superior olivary complex can be said to be the first place in the ascending pathway which receives information from both the ears. So this is the importance of the superior olivary complex. Now coming to the third order neuron. So obviously the third order neuron will arise from the superior olivary complex, enter into the lateral lemniscus and reach the inferior colliculus, which is located in the midbrain. Now, what about inferior colliculus? Now here, all the projections terminate here. So it is the site of convergence of most of the auditory information. So this is the significance of inferior colliculus. The superior colliculus, olivary complex is a place, first place where it receives the bilateral information from the bilateral ears, whereas inferior colliculus is the site where the convergence of the auditory information happens. So the fourth order neuron will arise from the inferior colliculus and go to medial geniculate body. Now, we always get confused between medial geniculate body and the lateral geniculate body. Means what kind of information it carries. So to simplify this, we can say music goes to medial geniculate body and light goes to lateral geniculate body. So music means hearing. So the medial geniculate body is concerned with hearing and the lateral geniculate body is concerned with light, that is vision. So music goes to medial geniculate body and light goes to lateral geniculate body. We come to the fifth order neuron. So the fifth order neuron will arise from the medial geniculate body, from where the auditory radiations will arise and terminate in auditory cortex. So we can say the auditory pathway can be summarized as from the cochlear nucleus towards SLIM. So S-L-I-M. You can remember this mnemonic. And then from here it goes to auditory cortex. So SLIM is nothing but S stands for superior olivary complex, L stands for lateral lemniscus, I stands for inferior colliculus and M stands for medial geniculate body. So this is how you can remember the auditory pathway. The auditory pathway, as it has got 5 orders of neuron, this formula may help you in remembering it. Now coming to salient features of auditory pathway. From the pathway, it is very evident that it has got bilateral representation. The second point is descending pathways. Third is non-lemniscal pathways, or other word for that is collaterals, which arises from the auditory pathway. Then it has got also a role in brainstem and spiral acoustic reflex, also known as attenuation reflex, spatial organization, and tonic map in auditory cortex. Now let us see these features in detail. A bilateral representation. Why bilateral representation? Because we have seen that the fibers have been crossing at different levels. So signals from both the ears reach both sides of the brain from medulla onwards. And preferably, the preference is towards to the contralateral pathway. So bilateral representation occurs because of the crossing over taking place at 3 places. So first is the trapezoid body is here. Okay, so the first point of crossing over is trapezoid body. Second is the lateral lemniscus. So in the lateral lemniscus, near the commissure between the both nuclei of lateral lemniscus, the crossing over takes place. And third place here you see is the inferior colliculus. So again, the third point is commissure connecting both inferior colliculus. So the crossing over takes place at trapezoid body, near the lateral lemniscus and the inferior colliculus. So because of the crossing over at these 3 different places, the information of one ear is sent to both sides of the brain. Coming to non-primary or non-lemniscal pathways. Now here, if you see the connection from the cochlea towards the spiral ganglion, and from here it terminates near dorsal and ventral cochlear nuclei. So here you see that some information is going to this pink structure that is nothing but reticular formation. Some information is going to cerebellum. From here, some information is also going to thalamus. So these all collaterals form non-primary or non-lemniscal pathways. So in short, we can say that auditory pathway is not only just concerned with hearing, it has got other different roles. So the collaterals to the reticular activating system, cerebellum, and non-specific thalamus or specific nuclei of thalamus. Now, as we know that the reticular activating system and the non-specific thalamic nuclei has got projections towards different parts of the brain. That's why activation, entire nervous system gets activated in response to loud sounds because of intense connectivity with the different parts of the brain. So what is the role of this collateral? First is role in arousal. That is, whenever a loud sound is heard, even if we are in deep sleep, we wake up. The best example is early morning when the alarm rings. We get up. Why? Because auditory pathway has got is sending collaterals to the reticular activating system, which will activate the entire nervous system. Second role is in motivation and attention. Now here, in spite of so much noise in and around, we do manage to read a novel in the train. So this is an example of role in motivation and attention. Third, the collaterals also play an important role in emotion and auditory reflex. The best example is attenuation reflex, where whenever you are exposed to a sudden loud noise, the ossicular chain becomes rigid and it protects our cochlea from effect of loud noise. So in short, we can say that collateral has got different roles. But during sleep, the link between the reticular and the wake and the motivation centers are inactive. That's why during deep sleep, we are usually not wakened up. Now, to summarize, this is the primary pathway, that is from the cochlear nucleus to the superior olivary complex, inferior colliculus, then to the medial geniculate body, that is the nucleus of the thalamus, and then to the auditory cortex. Also, because it has got collaterals to reticular formation, and from there it has got connections with the non-specific nuclei of thalamus, it plays a role in emotion and behavior. Now, spatial and tonotopic organization. What it means? It means that the conversion of the sound frequency to the coding is related to a specific position of excitement. It starts right from the organ of Corti, that is our hair cells, and this definite sequence is continued from the cochlear nucleus to superior olivary complex, inferior colliculus, medial geniculate body, and auditory cortex. Means, in short, at each and every level of the ascending auditory pathway, the spatial organization is strictly followed. Now, we know it that each part of the organ of Corti responds to different frequencies right from the basilar membrane to the apical portion. And why this occurs is because of the mechanical properties which vary at different portions of the basilar membrane. Now, at the base of the basilar membrane is thick, it is narrow and stiffer, whereas at the apex portion, the membrane is thin, it is more wider and compliant. That's why near the basilar membrane, if you see at the base, it resonates at higher frequencies, whereas at the apex, it responds to lower frequencies. So this pattern is followed very strictly in organ of Corti. Second, in the cochlea. So if you see here, this is what I was talking about, the hair cells. They respond, each hair cell responds to a particular frequency. Here again, you look here at the base, it starts right from 20,000, and as you move towards the apex, you reach towards the least frequency, that is 100. Now, in cochlear nucleus, if you see the arrangement, now apex, it corresponds to the anterior portion, whereas the base corresponds to the posterior portion. So, the apex, the anterior part of the cochlear nucleus responds to the lower frequencies, whereas the posterior part of the cochlear nucleus responds to higher frequencies. So, this type of organization is followed in cochlear nuclei. So, coming to inferior colliculi, again, here this section is the anterior and this is the posterior. So, again in the anterior portion, it responds to lower frequencies, whereas as you move towards the posterior, the section starts responding to higher frequency. So, this is the organization in inferior colliculus. Now, in the auditory cortex, now similar to the arrangement in the basilar membrane, the auditory cortex is also divided into 6 tonotomic maps, and they follow columnar organization. Why this type of mapping is important in auditory cortex? Because the auditory cortex has to do the job of discrimination of frequencies, and also it has to detect the direction of sound frame. Means it is a role of what and where, means knowing what kind of sound it is and from where it is coming. So these information is encoded in auditory cortex, that is what sound and from where it is coming. So the auditory cortex, we have the primary auditory area, this light green portion. Then we have the secondary auditory area. The posterior portion is known as the Wernicke's area, which plays a very important role in speech. And we have the Broca's area, also known as motor speech area. So what are the characteristics of this auditory cortex? As we know, our cerebral hemispheres are divided into right side and the left side, and most of the people are right-handed, means the dominance of hemisphere is towards the left. So our auditory cortex also shows marked hemispheric specialization, means in the dominant hemisphere, the Wernicke's area is more active. For example, in the right-handed people, the Wernicke's area in the left hemisphere is more active. And when it comes to right side, in the right-handed people, the right portion is dedicated to melody, speech, and sound intensity. So you can just remember this figure, that is the left side, which is involved in cognitive functions, that is logical thinking, calculations, whereas the right side is concerned with music, melody, pitch, and creativity. So another important characteristics of auditory cortex is marked neuroplasticity. Now what is neuroplasticity? Means continuously the neurons, they get adapted to encoding new information and storing new information. So our auditory pathways are very plastic and they are continuously modified by the experiences. The best examples for this are in the deaf person. Now the deaf persons, here the sign language activates the auditory cortex. So this type of plasticity is shown by the auditory cortex in deaf people. Now coming to blind people. Now blind people usually have better localization of the sound. So this is another example of neuroplasticity. And very commonly seen amongst the musicians that a larger part of the cortex is dedicated for audition and encoding of frequencies and music. So these are the different examples representing marked neuroplasticity in auditory cortex. Now, what are the connections in the auditory cortex? Now, as we know, we have the Broca's area and the Wernicke's area. The Broca's area and the Wernicke's area are connected by arcuate fasciculus. So this is another diagram depicting the flow of information, that is, whenever you hear any sound from the primary auditory area, because when you want to speak, that is language and speech go hand in hand. So from the primary auditory area, the information, the comprehension of the language occurs in Wernicke's area. From the arcuate fasciculus, that information will go to Broca's area, which is a motor speech area. Now with the help of the premotor cortex, the speech is delivered. That is articulation of the muscles, pharynx, larynx, everything occurs and you deliver speech. So what is neural processing of auditory information? Means whenever you hear a sound, the question arises is what type of sound it is and from where it is coming. So very commonly taught in kindergarten is like, you know, what does the duck, what kind of sound the duck makes, or the cow saying moo. So this is how from lower classes we are taught to identify what kind of sound it is. Even if your colleagues are talking blindfolded also, you can identify whose voice it is. So this is a property of auditory cortex. Second is from where. So along with knowing what kind of sound it is, we are able to sense and understand the sound. We are able to know from what distance the sound is coming, and also we are able to determine from which direction the sound is coming. Now how all this is possible? It is because our auditory cortex has got capacity of encoding the sound frequency. It has got capacity to know the loudness or the intensity of the sound. It can also detect the various features of the sound, and we can also locate from where the sound is coming, that is location of sound in space. So all these properties constitute the neural processing of auditory information. So let us see one in detail. Now what is encoding of sound frequency and pitch? Now we know that humans have got the audible range from 20 Hertz to 20 kilohertz. Now, so pitch means which neurons in the basilar membrane are responding to the sound. So it is a job of the cochlear nerve fibers to encode the frequency of sound waves. So based on this principle, we have what two theories, that is place theory or the Bexey's traveling wave theory, or the Volley's principle or frequency theory. So coming to traveling wave theory, now we know that in the basilar membrane, depending on what kind of frequency it is, the sound wave will cause oscillation of that particular section of the basilar membrane. For example, if it is a frequency of 20 Hertz, the base of the basilar membrane will be set into vibration. If it is 15 Hertz, the middle portion, and if it is around 20 Hertz, the apical section of the basilar membrane is set into vibration. So, the basilar membrane acts as mechanical analyzer of the frequency of sounds. The picture of this is George von Bexy. He had won Nobel Prize in 1961 for determining the physical mechanism of excitation of cochlea. He had put on theories about the mechanical properties of the basilar membrane. So coming to traveling wave theory. Now here you see again another diagram depicting the traveling wave theory. So this is 20 Hertz, 20,000 Hertz wave. So which gets completed here itself. So if it is 2000 Hertz, it will travel a bit longer across the basilar membrane and end at this section. If it is 500 Hertz, it will travel a larger portion of the basilar membrane and end towards the apex. So depending on the frequency, the distance from the stapes varies. So depending on which section of the basilar membrane is set into vibration, similarly, that area of the hair cells undergo excitation and send efferent impulses across the nerve fibers. So we can say that specific frequency sets into vibration of a specific location of the basilar membrane. Accordingly, that set of hair cells are excited, which sends information to the auditory nerve fibers. So this is the traveling wave theory. Coming to Volley's theory or frequency theory. Now, as we know, I have said before that in the cochlear nucleus also a specific tonotopic map is followed. That is, the anterior section responds to lower frequencies and the posterior section responds to higher frequencies. Now in Volley's theory, this is usually set for frequencies towards the lower section, that is less than 2000 Hertz. So this theory plays a role. Now here, Volley's means cohort or group of frequencies. Now, the lower frequencies of sounds are usually synchronized towards the same frequencies, means a group of same frequencies ranging between 200 to 2000 Hertz are considered as same frequency at the level of basilar membrane. But when the information is transmitted via the cochlear nerve towards the cochlear nucleus, the cochlear nucleus does the job of differentiating the frequency between 200 to 2000 Hertz. So it is at this level of cochlear nucleus where the differentiation of the lower frequencies would take place. Usually it is of a very limited importance. The most important theory is a place theory, but in the exam when the question is asked about the theories of hearing, you have to mention both of them. Now, how the loudness or the amplitude of the sound is encoded. Now, it is determined by the rate of responses and the number of neurons which respond to sound. So, look at this diagram. So, this is the single layer of the inner hair cells, and these are the outer hair cells. So, for example, this is a sound of lower amplitude which is reaching this organ of Corti. So the basilar membrane will be set into vibration, and these initial 3 rows would be of hair cells would get stimulated or excited. Now, when a bigger amplitude of same frequency of sound comes, means the same section of the basilar membrane will be set into vibration. But when you see the number of neurons getting stimulated, it is much more higher than the previous one. So, if the same frequency of sound but with higher amplitude means more loudness reaches the organ of Corti, it causes more firing of the hair cells. So, we can say that louder sound causes more vibration of amplitude in a larger scale, which causes more stimulation of the hair cells, or more of the hair cells are getting stimulated, which results in increased firing. So there occurs spatial summation of all these impulses leading to more activation of auditory fibers. Usually, our outer hair cells do not get stimulated for lesser amplitude of sound, but whenever the outer hair cells get stimulated, it means that there is a loud sound. So, stimulation of outer hair cells apprises the nervous system that the sound is very loud. So, whenever the high intensity of vibration sets basilar membrane into vibration, the outer hair cells get stimulated. Coming to features of sound waves. Now, as we know, sound waves have got different features. That is intensity, speed, all those things. So all these are also responded by the cortical neurons. For example, in this diagram, the sound waves are reaching the ears at higher frequencies as compared to this diagram, where the sound waves are reaching at a lower speed. So the central nervous system has got a capacity to differentiate between the intensities of sound reaching the two ears. As you know, the head forms a barrier between both ears. So there's a lag occurring the sound when it strikes the right ear and then the sound strikes the left ear. Similarly, over here. So CNS has got the ability to detect this time difference or the delay which occurs between the arrival of sound between the two ears. Now, localization of sound in space. A very common example is if you forgot your mobile phone, okay, not in vibration mode. In the ringing mode, if you forget where you have kept your mobile phone, so what we do, we take our friend's phone and we give a ring and we try to search wherever possible. How are we able to find our phone? Because we are able to locate from which direction the sound is coming. Let us see how it is possible. Now, here, this is known as localization. So, for example, this is an aeroplane which is flying. It will cause sound waves to reach the ear. But if you look at the length, this side of the ear is lesser one and this side is more. So, there is always a time lag occurring between the entry of sound in both the ears. Now, this is reflected as number of impulses. If the sound reaches the right ear earlier, it will set this, it will cause more vibration from the right ear, whereas there is a lag of the sound waves reaching the left ear, so there will be delayed firing of those hair cells. So that's why we can say if a sound source is perpendicular, then the sound waves will arrive simultaneously on both the ears with the same intensity. So here the shape of the pinna will change the quality of sound entering. Either it is from the back side or from the front. So this is how we are able to determine whether the pinna helps in determining whether the sound is coming from back or from the front. So the shape of the pinna helps in this process. If the sound is coming horizontally, so as the head acts as a barrier, there is a time interval. So difference in these acoustic signals helps in determining from which direction the sound is coming. Either it is coming from the left side or the right side. So this differentiation begins in superior olivary complex. For example, here, the fire truck at rest. So as the fire truck is nearing, the intensity of the sound goes on increasing. So here the lateral nucleus plays a role in determining the difference of intensities of sound waves reaching both the ears. Whereas here, the medial nucleus helps in determining the time lag between two acoustic signals, that is for the horizontal sound. So because of this, a set of neurons are set into vibration earlier and later. So this difference of time lag is detected by medial nucleus. So difference of intensities is a job of lateral nucleus, whereas the time lag detection is a job of medial nucleus of superior olivary complex. So localization It is a superior olivary complex which plays a very important role. So this is how the spatial orientation of the signals leads to localization of sound by auditory cortex. Thus, the quality of sound of direction is separated from the quality of sound tones at the level of superior olivary complex. So, listening and hearing, are both of them same or different? Now, there is another property of the auditory pathway is the centrifugal signal. The centrifugal signals are nothing but some projections arise from the auditory cortex and retrogradely sent bilaterally into every level of the central auditory system. Now, as you see in this diagram, there are projections going to medial geniculate body, inferior colliculus, cochlear nucleus, superior olivary complex. So auditory cortex is also in connection with different nuclei of the auditory pathways. So we can say this whole complex is highly interconnected. Now this descending feedback, it depends upon the alteration and the importance of the environmental and attentional fluids. Now let us just revise a bit about the outer hair cells. Now the outer hair cells sends 10% of afferents and receives 90% of efferent information. Now the outer hair cells have got pristine, that is a motor protein located in the cell membrane. Now this pristine offers somatic motility in parallel to the changes in the potential occurring in the outer hair cells. So whenever the potential changes, there occurs configurational changes in the pristine molecule, because of which the length of the outer hair cells change. So whenever depolarization occurs, the outer hair cells shorten, and whenever hyperpolarization occurs, outer hair cells lengthen. So why this is important? Because whenever sound is arriving at the basilar membrane, so in response to loud sound, the outer hair cells will get stimulated. So when they get stimulated, they will get shortened, that is in response to depolarization. So whenever they shorten, what is going to happen? This basilar membrane will become stiff. So stiffness of basilar membrane means it will respond less to the sound which is arriving at the cochlea. So because of which it dampens the excitation of the hair cells. Thus, it is a protective mechanism. So length of the outer hair cells helps in adjusting the sensitivity of the inner hair cells. So we can say from the superior olivary nucleus, the bundle which connects the hair cells is the olivocochlear bundle. Now, olivocochlear bundle is usually inhibitory in nature. The inhibitory nature is because of the release of acetylcholine at the level of hair cells. Thus, we can say that this cortical feedback causes conformational changes in the outer hair cells, which helps in protection of the hair cells. Also, it plays a very important role in the environment where we are exposed to loud sounds. So, direct stimulation of discrete points in the olivary nucleus helps in inhibiting specific areas of organ of Corti, because of which there is reduction of intensity by 15 to 20 decibel. The best example is in party. In and around there is loud sound, but still we are able to converse with each other. So this ability is because of the centrifugal signals which are arising from the auditory cortex and inhibiting at different levels. That's why I say you hear what you want to hear. We usually tend to ignore. So this ignoring capacity is possible because of this centrifugal signals which are arising from the auditory cortex. So the difference between listening and hearing is, hearing means it is perception of sound, means what sound is you're hearing. Whereas listening means understanding the auditory information. Hearing is a passive process. Whether you want or you don't want, still you will hear the sound. But Listening is an active process. Means hearing involves effortless activity, whereas listening involves conscious efforts. It needs conscious efforts. It also needs interest and concentration. That's why listening leads to learning. So to conclude, we can say auditory pathway, it starts right at the level of hair cells, which are located in the cochlea, which goes to superior olivary complex, lateral lemniscus, inferior colliculus, and medial geniculate body. And from the medial geniculate body, the acoustic radiations project into the auditory cortex. So this is the summary of auditory pathway. Now this auditory pathway has got different salient features. That is bilateral representation. That is the information from one ear can reach both sides of the brain because of crossing over taking place at 3 points. That is at the trapezoid body. Second is at the nucleus of lateral lemniscus. And third is at the level of inferior colliculus. So the crossing over of the fibers help in bilateral representation of the auditory information. That's why lesions above the cochlear nucleus do not cause major hearing impairment. Now, the auditory pathway also has collateral pathways to the reticular activating system, to the non-specific nuclei of thalamus, and also to the cerebellum. So because of these collateral pathways, the role of this is in arousal, in emotion, in attenuation reflex, and also in attention. Now, centrifugal fibers play a very, very important role in dampening of the inner hair cells excitation whenever you are exposed to loud sound. And also it helps in attention behavior. Right from the level of organ of corti till the auditory cortex, it follows the rule of spatial organization. This ability of spatial organization is important for discrimination and detection of direction of sound. The direction of sound is attributed to the superior olivary complex. We have also seen the theories of hearing, that is the place theory and the Volley's theory. Now, place theory was described by George von Wesecki. Now, according to the place theory, different sections of the basilar membrane responds to different frequencies of sound. Whereas the Volley's theory plays a role in determining the differentiation of frequencies less than 20 Hz. Here, the cochlear nucleus plays an important role in differentiating the different frequencies. And the last is the neural processing of the auditory information helps in encoding the frequency and intensity and features and sound localization. Thank you very much for your patient listening.